Thank you, gang, and thank you to everyone again for joining us today. Turning now to answer the question, why microneedle array patches? When it comes to conventional injections versus MAPs, in terms of patient experience, injections can be painful and drive needle phobia. MAPs are designed to be painless, improving acceptance and adherence. When it comes to administration and access, injections typically require trained staff in clinical settings, whereas MAPs enable community-based or even self-administration. For safety logistics, injections create sharps waste and needle stick risks, but MAPs eliminate sharps, providing for safer handling and disposal. In terms of performance for vaccines and biologics, muscle tissue is not as rich in immune-competent cells, and biologics often require chronic injections. Skin access to immune cells via MAPs can improve bioavailability and dose efficiency for vaccines and biologics because of direct access to dermal immune cells and by bypassing the GI respectively. For scalability and outbreak response, injections require healthcare infrastructure and cold chain, while MAPs can potentially avoid the cold chain and supports rapid, scalable deployment. In terms of commercial differentiation, conventional injections are often of mature products which face competition and loss of exclusivity. MAPs can provide an opportunity for lifecycle management and product differentiation. In short, the answer to the question of why MAPs is they offer a patient-friendly, scalable, and biologically effective alternative to traditional injections, especially for vaccines and biologics. MAPs have a broad range of applications, from vaccines including mRNA for flu, COVID-19, HPV, measles, and pandemic stockpiles, to peptides and hormones like GLP-1, insulin, parathyroid hormone, and hormones for contraception, to proteins and non-monoclonal antibody biologics like cytokines, growth factors, and for wound healing. MAPs can also be used to deliver oligonucleotides like DNA and siRNA for vaccines and gene silencing, as well as small molecules to treat pain and inflammation and CNS diseases. Diagnostics for biomarker sampling and aesthetics for anti-aging and hair loss are also appropriate for MAPs. And MAPs fit for these because of the possibility for dose sparing, strong skin immunity, potential thermostability, painless chronic dosing, predictable PK, and minimally invasive administration and sampling. While there are many applications for MAPs in those categories, I would like to further refine what are ideal and not so ideal to be delivered in MAPs. MAPs are suitable for delivery when the drug benefits from being in the intradermal space. If the dose required is below about 3 milligrams, the API is water soluble, though we have ways around this, cold chain avoidance adds value, and self-administration or multi-dose adherence matters. MAPs are not ideal if the drug is already effective via IM or oral delivery, the required dose is in the high milligram range, the API is insoluble without enabling strategies, there is no benefit from avoiding cold chain, in single dose use cases, or with ultra-low cost generics. Here is an overview of microneedle designs. First are dissolving microneedles, also known as poke and dissolve, where the drug is embedded and formulated as part of the needles, which then dissolve in the skin and then release the drug. Solid removable microneedles are known as poke and patch. Here the solid needles make tiny holes in the skin so that the drug applied later onto the skin can then fill the holes made by the microneedles. Coated microneedles, also known as coat and poke, have the drug coated on the outside of the solid needles, which then dissolves into the skin after insertion. Hollow microneedles, or poke and flow, act like mini injections whereby the drug is pushed through the skin through narrow channels in the needles. Hydrogel forming needles, or poke and release, have a drug reservoir above the needle matrix. The microneedles swell in the skin to slowly release drug from the reservoir, or in the case of biomarker sampling, collect the fluid into the reservoir. Each design has its pros and cons with respect to drug load, release, manufacturability, and waste profile. LTS chose dissolving microneedles as the basis for our AccuTip platform. Reasons for this are that they enable optimal API loads without external reservoirs, support immediate or sustained release options, stabilize APIs within polymers, have minimal skin residence time compared with classical transdermals, and offer flexibility in needle geometry and composition. I would also like to note that we have partnerships using our CMO services that work with hydrogel needles. Here's a closer look at what happens in the skin with these dissolving microneedles. After insertion into the skin, the polymers dissolve and release the API over minutes, after which the backing is removed with no sharp edge waste. As previously mentioned, the dissolving microneedles can accommodate a medium drug load of up to about 3 milligrams, offer polymer choice flexibility, have a short lag time comparable to injections, have availability of immediate and sustained variants, have no sharp edge waste, and have a short application time on the skin. Now, more specifically, I'd like to talk about LTS's MAP program. We have both our proprietary AccuTip platform as well as contract development and manufacturing to bridge from concept to clinic and beyond. We're structured to move from feasibility to clinical supply and set the stage for commercial scale. Some years back, we did a gap analysis on the microneedle market. This is a map of MAP developers from PATH from a few years ago. Here we see that there are many startups and universities, but there is little to no industrial production. LTS's MAP program can fill the gap from bench to patient by aligning process, scale, and regulatory readiness. LTS's MAP program scope covers the value chain, from feasibility to formulation and process development, to clinical support, and finally large-scale manufacturing. Our role is to be a trusted development and scale-up partner through commercial readiness and supply. Our MAP program is organized like the broader company, a business-to-business CDMO and drug delivery enabler. In the innovator, we are focused on formulation development with our AccuTip platform with a partner's API. We execute screening programs, extended formulation work, provide preclinical and clinical samples, and can do device development should an applicator be required. In our CMO operation, we focus on larger scale manufacturing. Here we can provide process development, upscaling, or tech transfer for partners that have come from the innovator or other partners with their own MAP platforms who may not have the GMP, QA/QC, or larger scale manufacturing infrastructure. Finally, borrowing from the capabilities of the larger LTS organization, we are able to tap into our engineering department should additional machine development and construction be required, and our analytical departments for additional support in this area. In this next section, I'll focus on LTS's MAP Innovator and our AccuTip platform. We'll touch on the technology, portfolio, proprietary data, and manufacturing differentiators to support partners. First, the AccuTip technology. Here is a standard AccuTip array packaged in a blister pack. It is 1 centimeter in diameter, 0.8 centimeters squared in area, 600 micrometer needle length, and there are 300 needles in this array. We can customize length, width, density, and composition to match program needs. So now that you know a little bit about the AccuTip patch, I want to give you an overview of the various programs we have ongoing with partners with the platform and the multiple modalities we have to be able to deliver with AccuTip. The LTS AccuTip portfolio spans development through clinical across various modalities. We are working with partners developing vaccines with mRNA, LNP, live virus, virus-like particles, proteins and peptides, and small molecules. We also have programs with partners to deliver biologics as proteins and peptides, antibodies, small molecules, and even for aesthetics. Now that I've shown you the different modalities we are working with in our AccuTip platform, I will now show some data related to a few of those modalities in both vaccines and biologics. A disclaimer before I go through the data. As a CDMO, LTS is bound by our contractual obligations to protect partner confidentiality. So in order to demonstrate the capability of the AccuTip platform and be able to show the data, LTS executed its own internal proprietary programs, namely a phase 1 hepatitis B surface antigen virus-like particle vaccine booster study, a collaborative preclinical study with mRNA LNP encoding rabies glycoprotein in guinea pigs with CureVac, where they gave us permission to share the data, and a preclinical study with interferon beta in mini pigs. To start, here is the phase 1 hepatitis B surface antigen vaccine booster study we performed. The molecule is the surface antigen of the hepatitis B virus. It is 22.5 kilodaltons in size and arranged in virus-like particles, which means it's actually a pretty big molecule and upwards of 3 megadaltons. The vaccine was produced in Hansela cells, and we used the bulk material provided by the Serum Institute of India, which they typically use to make the commercial hepatitis B vaccine, also known as Ingerix B. However, the bulk was provided to us for the Microneedle Array Patch in this study, and it did not contain adjuvant like the commercial product. The clinical trial study design was a booster study. We recruited healthy male and female volunteers who were successfully vaccinated in the past. We set a tighter range in which the subjects had to fall. We had a lower cutoff of 10 international units and an upper cutoff of 400 international units. We set the lower cutoff because particularly with hepatitis B, there is a certain percentage of the population that does not respond to hepatitis B vaccination. Thus, in order to rule out that it was the MAP vaccination that did not result in increased titers versus the subject just wasn't going to respond, we excluded that part of the population with titers lower than 10 international units. We set the upper limit to make sure we could see a twofold increase required for a booster. It was a randomized, open-label phase 1 study. We took enrollment titers at day -14. The T0 value was taken at day 0 to get the baseline titers, and then the participants were given the MAP booster. We asked them to come back at day 31 to collect samples for the boost titers, and we had a follow-up at day 180 for safety measures. We had 2 trial arms, one using an applicator device to administer the patch, which was compared to the second arm, which was a manual application. The reason for these two arms is that we wanted to see whether or not an applicator is necessary, or if manual application would be sufficient. Not needing an applicator for microneedle patch products would be an advantage over conventional injection. We had 20 volunteers per arm, and each of them received 20 micrograms of antigen per application, which is the equivalent of the commercial hepatitis B vaccine. In this case, we had to apply 2 MAPs with 10 microgram each antigen since we received bulk material which was not up-concentrated. In this picture, you see the clear over patch here, and the red circle is the microneedle array. For this study, we had to fix onto the subject the patch with over tape because we were asked by the health authorities to leave the patch on for 24 hours to look at worst case reactogenicity. In a real product, this would not be necessary, and the patch could be removed in 10 to 15 minutes. After removal, you can see where the actual microneedle array was located and a little redness at the site, which is to be expected given a vaccine was administered, but no significant reactogenicity, and the redness was resolved within 24 hours after patch removal. The application was very well tolerated and described as completely painless. There were no adverse or severe adverse events reported even through the 180-day safety check. The average baseline antibody titer upon enrollment for both arms was about 85 international units per liter. With the manual application, we were able to boost titers to an average geometric mean of 1,120, which is about a 14-fold increase over baseline. And with the device to almost 11,000 international units per liter, which is a 116-fold increase. Compared to injection of the Adherence B commercial product from data from a previous study were performed, you can see that even with manual application, MAP titers on average were better. And to reiterate, the hepatitis B vaccine in the MAP contained no adjuvant, unlike the Adherence B that was used for the injection. In summary, MAP vaccination induced a strong immune response with and without an applicator and had excellent tolerability. The immune response was also better than the IM injection without having an adjuvant. The key takeaway here is that the MAP approach can meet or exceed intramuscular benchmarks with an improved patient experience. Now shifting to work LTS did in collaboration with CureVac a handful of years ago. Given the high interest in mRNA vaccination in the past and challenges with ultra-cold chain storage, we wanted to develop a microneedle array patch containing mRNA encapsulated in lipid nanoparticles, or LNP. We received mRNA encoding rabies virus glycoprotein based on CureVac's proprietary second-generation backbone with optimized non-coding and coding regions encapsulated in CureVac's proprietary ionizable lipid with distinct features. Guinea pigs were the animal model used for this prime boost study. mRNA LNP MAPs were stored at 5 to 8 degrees centigrade in the refrigerator, while bulk material was stored in a -80 freezer. Animals were shaved on day 1 prior to MAP application or injection, and the MAP or injection occurred at day 0. Blood was collected 1 day after prime MAP application or injection of the bulk material. On the day of the first application, we noticed hair growth after 24 hours of up to 1 millimeter in length. As the microneedles are only 600 micrometers in length, this resulted in incomplete insertion of some of the microneedles. From our experience with the hair growth during the prime vaccination, animals were shaved on the same day just prior to the boost MAP application or injection at day 21. Blood samples were also collected on day 21 as well as day 35. There were 8 guinea pigs per arm, MAP, bulk intramuscular injection, and untreated animals. The vaccine titers after prime application of MAP, which was stored in the refrigerator, or bulk material stored at -80, showed protective titers. The lower titers for the MAP application are likely due to the hair growth in the animals preventing all needles from penetrating and thus an underdosing of the mRNA. We observe a significant increase in titers after boost application of both MAP and bulk injection. The boost titers are still lower quantitatively between MAP and bulk material. However, the fold increase from prime to boost titers is higher with the MAPs. We observed after the modification of the shaving of the animals the same day of boost application of MAPs that all needles were allowed to penetrate. We postulate that if this had been the case for the prime application, vaccine titers for the MAP application would have been at the same level as that of the injection. In all, this demonstrates successful vaccination of guinea pigs with mRNA encoding rabies glycoprotein, incorporated in a microneedle array patch. In practice, for human application, we should be cognizant of this and target areas like the deltoid or inside of the forearm without hair to apply microneedle array patches. Next, we sought to evaluate the stability and integrity of mRNA and MAP for a prolonged period of time at elevated temperatures. We used mRNA encoding luciferase encapsulated in LNP provided by CureVac. In blue, you will see data corresponding to MAPs that were stored in the refrigerator at 5 degrees. In green, data for MAPs stored at room temperature at 25 degrees. Red are data points for MAPs stored at elevated room temperature of 40 degrees. And finally, the open diamond and dotted black line is the bulk mRNA LNP solution not formulated in a MAP and stored in a -80-degree ultra-low freezer. LTS has established and validated the required analytical methods to perform the characterization required of mRNA LNP containing MAPs, such as particle size determination by dynamic light scattering, multi-angle light scattering, and flow field fractionation. We determined mRNA encapsulation by performing ribogreen fluorescence assays and see that the mRNA remains encapsulated at all temperatures post-formulation for 12 months and is comparable to the bulk material. mRNA integrity was analyzed by microcapillary electrophoresis, fragment analyzer, and HPLC. We see that mRNA integrity can be maintained up to a year with MAPs stored in the refrigerator, though rapid degradation is observed at higher temperatures. We determined mRNA transfection efficiency using cell culture assays. We see high transfection efficiency is also maintained over 12 months with MAPs stored in the refrigerator, even better than the bulk material which was stored at -80. In summary, MAP vaccination with mRNA LNP induced protective titers. We were able to show successful incorporation of mRNA LNP in MAP, and we were able to elevate storage temperature of mRNA LNP in MAP from -80 to 5 to 8 degrees centigrade for over a year, demonstrating improved thermal stability. LTS also ran a proprietary study with recombinant human interferon beta 1a. The molecule has a molecular weight of 22.5 kilodalton, so it is a relatively large molecule. It is produced by CHO cells with the human interferon beta gene and the amino acid sequence of the recombinant protein identical to that of the natural human interferon beta. The indication of the marketed recombinant interferon beta is multiple sclerosis for patients with slow disease progression or relapsing disease. The commercial products Avonex from Biogen and Rebif from Merck have been on the market for quite some time. Despite this, sales were still over $1.5 billion last year. We chose this molecule as a proof of concept because of the chronic dosing and lifelong nature of this medication, which, as previously mentioned, is an area where MAPs would be appropriate. Patients are relatively young, and providing a painless application alternative which would not interfere with their general lifestyle would be preferred and beneficial, especially with respect to improved therapy compliance and adherence. For the preclinical study, we used Göttingen mini pigs. We had a high-dose group of 5 animals receiving the equivalent of 150 micrograms of interferon beta, and a low-dose group receiving the equivalent of 30 micrograms interferon beta, which is the dose of the commercial product. We also administered intramuscularly and intradermally 3 animals each with 30 micrograms of Avonex for comparison. As you can see, we took blood samples a day before administration and then at certain time points after, measuring the amount of interferon beta in the plasma. The results of the study were published last year in the Journal of Drug Delivery Science and Technology, but I will give you a brief summary of the key findings now. On this slide, we show the comparison between the intradermal and intramuscular injections of 30 micrograms of the marketed Avonex product in the mini pigs. As you can see, there is a comparable area under the curve, whether intradermally or intramuscularly injected. On this slide, we took that intradermal gray curve and placed it here to compare to the intradermal delivery via MAP of the 30 microgram equivalent or 150 microgram equivalent. As you can see, the 30 microgram equivalent achieves a comparable area under the curve as the 30 microgram intradermal injection of the Avonex product. Then when compared to the 150 microgram MAP equivalent, we see a dose correlation of about 5 times the interferon beta in the plasma. This shows that the MAP technology in a larger preclinical model can deliver interferon beta comparable to an intramuscular or intradermal injection of the marketed product. In addition to biologics, LTS is further working on delivering recombinant protein subunit vaccines with partners. I've described the Accutip platform and technology, what actives we've worked with, shown you shareable data in the realm of vaccines and biologics, and now I would like to tell you about LTS's differentiators in terms of microneedle array patch manufacturing. Here is an overview of the steps in the manufacturing of our Accutip MAPs. The first step is always the preparation of the dispensing solution. Here we use the expertise, experience, and knowledge we have in formulation for our other platforms as well as in the equipment that we use. The next step is dispensing the solution into the molds, followed by the drying step where the solution dries and the microneedles are formed. The dried microneedles are then demolded. The array is overlaid with a cover patch. We merge the cover patch with blister packaging, which then protects the microneedles from physical damage. And then we seal this into a packaging material to protect it from environmental factors like humidity, moisture, and oxygen. As mentioned earlier in this presentation, the LTS MAP program can fill the gap from bench to patient by aligning process, scale, regulatory readiness. Here are some of the key differentiators we have to address those gaps. First, LTS holds a manufacturing authorization from our local German health authorities for clinical MAP sample manufacturing. We are regularly inspected for GMP compliance, and these early interactions with a regulatory body helps LTS to address potential regulatory pitfalls in advance, which ultimately benefits LTS partner timelines to market. Second, all of the materials, including polymers, excipients, and molds, used by LTS for MAPs are GMP grade and suitable for parenteral application. This avoids extra regulatory pathways, saving money and avoiding delays to market. Third, the manufacturing process used to produce lab samples are already manufactured such that it can be transferred to large-scale manufacturing, which is designed for high throughput efficiency. Finally, LTS has a proprietary single-tip multi-layer dispensing technology, which I will describe a bit more on the next slide. For early development phases, we manufacture what we refer to as a monolayer microneedle array patch, because these allow us to move into preclinical and clinical studies fairly easily and quickly. We pour the dispensing solution into the molds, filling both the needle cavities and the base plate with API-containing polymers. Then, as described earlier, the solution is dried, the array is demolded, and the cover patch with the adhesive and backing layer is placed, followed by the blister packaging. We have learned from internal studies, however, that only 2/3 of the tips penetrate into the skin. So only the API in that part of the tips will be released into the body, whereas the upper part of the tips and base plate is just removed after the application and will not dissolve in the skin. Thus, moving to later phase development with LTS's commercial mindset, we know we cannot use this monolayer microneedle array, since this would not be acceptable to regulators who would question the dose accuracy of the application. This is where our single tip dispensing technology comes in, and one of the manufacturing differentiators LTS can offer our partners. With this dispensing technology, we can load 2/3 of the tips with the API-containing polymer, followed by the same polymers but without API for the upper part of the microneedles and the base plate, with the same adhesive backing layer and blister packaging. The advantages of this multi-layer microneedle array patch are that a dose sparing can be achieved, since only the part of the microneedle tips contacting the body are actually filled with API. Dose accuracy is also realized, since even if more of the needle tips get pushed into the skin, anything above the API-filled tips will not contain API. Finally, in the case of vaccines, if there is an adjuvant that works, but perhaps is not compatible pre-mixed with the vaccine, or in the case of multivalent vaccines, or with peptide pools wherein certain peptides do not interact well with each other in solution, we can individually dispense these tips with different actives, separating them in the different tips such that they do not counteract each other prior to reaching the intradermal space. We believe LTS can leverage these manufacturing differentiators and other capabilities to be a process development and manufacturing accelerator for companies, startups, and universities. We have the state-of-the-art GMP R&D center supported by our QA and QC system, where we can make early phase clinical samples under GMP with a dedicated microbiological lab, where we can handle even live viruses and highly potent APIs. We have regulatory experience with all the major agencies and have had scientific advice meetings with and are regularly inspected by our German health authority. We have an established network with respect to pharmaceutical manufacturing, academia, and industry partners, and access to equipment makers and raw material suppliers. We have a pre-pilot line successfully established with a concept for aseptic and sterile manufacturing. All in all, this suite of capabilities is what differentiates LTS from most other map developers. Turning now to our CMO operations. Here, we focus on the future production of maps for partners working with our Accutip technology from the innovator side, but we can also onboard third-party map technologies and act here as a contract manufacturer for the third party's maps technologies. Under our CMO operations, we focus on process development, either upscaling or performing a pure tech transfer. And by working with us for GMP manufacturing, a partner's time to market can be accelerated due to LTS's already established organizational structure and experience with GMP manufacturing. With respect to LTS's partners, you can see through our pipeline that we have a number of partners who have come to work with LTS directly to our CMO operations with various modalities, namely proteins and peptides, antibodies, small molecules, and in diagnostics. In summary, with respect to the present and future of map manufacturing at LTS, we have a pre-pilot line for GMP manufacture of dissolvable maps. Our novel single-tip dispensing system, which dispenses each array in less than 15 seconds, has been established. We have the capacity to support up to phase 2 clinical supplies. We are in the planning stages for a pilot manufacturing line that can produce phase 3 clinical supply, followed by the full commercial line for 10 to 20 million units per year per line, and the infrastructure planning for the MAP manufacturing site.